If you're in Monday lab, please make sure you notice Dr. Morgan's note up there about checking your transformants today. He said that got good results from the first lab section. Okay. Everybody get the email I sent out yesterday about the optional points activity coming up? It should be a good talk, so I would encourage you to attend if you can. Any questions? Okay, we have finished up photosynthesis, so my recommendation to you is that if you haven't done so already, you really start working on the study guide questions and kind of nailing down photosynthesis. But don't forget about that stuff now that we're starting a new topic, and I would encourage you to go back and occasionally brush up on the photosynthesis. Has anybody looked on the syllabus? What's our next exam? 2 weeks from Monday? 2 weeks from yesterday? Okay. So again, it will probably be 2 chapters on the exam. It will be photosynthesis and definitely DNA. Might get into a little bit of the next chapter. We'll see how things go within the next couple of weeks. So, what's so important about chromosomes? Why are chromosomes of interest to biologists? Areas of interest, certainly.
If you have a good understanding of chromosomes, you can understand a lot of diseases like Down syndrome.
Okay. Understanding of chromosomes, at least today, allows us to get some insight into diseases that have a genetic basis, like Down syndrome, so chromosomal abnormalities is interesting. What else do chromosomes do that's interesting? They can replicate. And when do they replicate? Okay, prior to both mitosis and meiosis, the chromosomes replicate. Did you talk last semester about the cell cycle? Okay. During the cell cycle, we break up the life of a cell into various events and represent it as this cyclical process where we have various phases, including a G1 phase, an S phase, a G2 phase, and an M phase for the actual mitotic division. So which of the phases of the cell cycle does DNA replication occur in? I'm going to go back. Yeah, there's a phase called the S phase that precedes the actual mitotic division. Okay? So chromosomes replicate prior to mitosis and meiosis. That's interesting. What are chromosomes? Okay, they are structures within the cell that are composed of 2 types of molecules. And those 2 types of molecules are DNA and proteins. And an important type of protein associated with DNA are proteins called histone proteins, which we'll talk about later on in this discussion of DNA when we start talking about some regulation of gene expression. So prior to cell division, the cell goes through some preparatory steps to get ready to have the DNA replicate. And then during this M phase of the cell cycle, these structures, these chromosomes, actually physically separate or segregate. Remember that they attach to that mitotic spindle, and that mitotic spindle pulls apart those replicated chromosomes apportioning them so that each cell gets the right number and right kind of chromosomes. So, they replicate prior to mitosis and meiosis. Well, how do they replicate? That's an interesting question. As Brittany implied, these units called chromosomes also contain information for genetic traits. One of the things you talked about last semester was the difference between the concept of a genotype and a phenotype. What do those two words mean? What does phenotype mean? It's an observable trait, okay? In the context of Mendel's original experiment, he looked at some easily observable traits which usually occurred in contrasting forms that were easily differentiated from one another, like a plant being tall versus being short, or having purple versus white flowers, or having yellow seeds versus green seeds. So those kind of outward manifestations that you can observe fairly easily, like eye color, hair color, are what we refer to as the phenotype. Basically, the bottom line is that a phenotype of an organism is really determined by the proteins that it produces. And so whether you have a purple flower or a white flower really is determined by, do you have particular enzymes that allow you to make those pigments or not make the pigments? Pea flowers that have the purple color have an enzymatic metabolic pathway and all of the necessary enzymes controlling that pathway to synthesize this purple pigment. White pea plants have some kind of a defect in that metabolic pathway. We have one or more enzymes that aren't working properly, and they don't make the pigment. And that absence of pigment is manifested as the white phenotype. Okay? If you have cystic fibrosis, you have a phenotype that involves a slightly altered membrane protein, which is very involved in the transport of chloride ions across the cell membrane. And if you have a mutant protein that codes for a slightly different chloride transporter, that can manifest itself in that particular condition of cystic fibrosis. If you have the normal protein, then it transports chloride normally, and you do not have that genetic disease. So what's genotype refer to? Which alleles of a gene you have, okay? So in Mendel's case, if P is the dominant allele for the purple phenotype, and lowercase p is the recessive allele for the white phenotype, then there are 3 different genotypes for that allelic pair, right? Homozygous dominant, homozygous recessive, and heterozygous. How many phenotypes are there? We have 2, right? Those would both be purple, and this would be white. So the genotype of an individual definitely does determine its phenotype. But again, the question is, how does that happen? Okay? How does having certain alleles allow you to make certain proteins that then determine certain traits? That's something we'll spend a fair amount of time talking about. So those are some interesting questions that scientists have kind of grappled with regarding chromosomes for quite some period of time. How do they replicate? How do they carry information for genetic traits? How do they encode information? So these are questions of function, right? And what's a good way for us to understand and dig into questions of function? To understand structure. Because if you remember one thing from biology, it is that structure determines function, right? So, prior to 1953, what did we know about chromosomes? I hate to date us, but we were both born before 1953. And so what did we know about chromosomes when we were really, really little? Not a whole lot. We knew that they were made up of two types of molecules, DNA and proteins. We knew that they replicated prior to mitosis and meiosis, but we didn't know how. We knew that they encoded genetic information, but we didn't know how. So there were a lot of unanswered questions about chromosomes prior to 1953. Why did I pick 1953? What's so important about 1953 when we start talking about molecular biology and molecular genetics? This is one of the dates that I think is very important to understand. This is really the kind of starting point for the work you did in lab yesterday, or will do in lab this week, with genetic modification of organisms. Samantha?
Isn't it when Crick and Watson, the DNA structure?
Mm-hmm. In 1953, James Watson and Francis Crick published one of the most important papers in biology ever, and probably without a doubt the most important paper in the field of what would become molecular biology and genetic engineering, in one of the most prestigious scientific journals of the time called Nature. The paper was actually part of a 2 or 3 paper set that was published simultaneously. And we'll talk about some of the other people who contributed to these other articles. But together, they kind of heralded the beginning of biotechnology, molecular biology, and our whole application of using DNA to modify organisms. Watson and Crick's paper was very, very simple. It was actually a single-page paper, and yet it had tremendous impact. I have a copy of that paper posted on our cell biology website, and I would really urge you to read it because I think it's kind of important to understand the origins of what we're going to spend the rest of the semester really digging into and what you're going to apply to your laboratory work. And so I think that this is really kind of foundational stuff that everybody should look at least once. So, prior to 1953, we knew that chromosomes were made of DNA and protein. One of the things that we didn't know, however, was which of these two components actually encoded the genetic information. At the time, most researchers favored protein as being the most important part of a chromosome. And they thought that the DNA part of the chromosome was this minor kind of structural molecule, but that the real kind of meat of the chromosome, the part of it that really encoded genetic information, was the protein. Any idea why the smart money at the time was on protein as kind of the encoding part of DNA?
Because proteins are more complex in their structure than DNA.
Now, proteins are a much more diverse set of molecules, okay? Both DNA and proteins are polymers, right? What is protein a polymer of? Amino acids. What's DNA a polymer of? Nucleotides, okay? How many nucleotides are there? How many amino acids are there? About 20, approximately 20. So if you're thinking to yourself, okay, this thing, a chromosome, encodes genetic information. That's got to be pretty complicated to encode genetic information. What's more likely, the portion that has kind of 20 variants or the one that has 4 variants? Well, you would think logically the one that had the more variants. more ways to encode complex information with 20 subunits versus 4. Here's something important I want you to understand about science. Just because something seems logical doesn't always mean that's the way it really is. That's what's true. A great example of that is photosynthesis. When scientists first study photosynthesis and we're trying to figure out, okay, how does this really work? We know what the reactants are. We know what the products are, but how do you get from here to here? Well, a lot of people said, well, okay, let's look at this logically. If carbon dioxide were somehow split, what does that leave you as a product? Oxygen, right? And what can happen to that carbon? The carbon could attach to water to make carbohydrate. Makes perfect sense, right? But in reality, is that what happens? No, when they started doing some experiments in photosynthesis and they started tagging some of these atoms with radioisotopes that they could then follow and see where they went, what they really found out was that it was the water that was split. Okay? So just because this made sense doesn't mean that's actually the way it worked. Well, that's really kind of what happened with protein as well. We thought originally that protein made more sense as a molecule that could encode complex genetic information. But in reality, it is the DNA that really encodes the instructions that determine phenotype of organisms. So, we're going to go through a little bit of history of science in seeing how all of this understanding of DNA developed. The first thing we're going to talk about is what kind of experiments that were done that nailed down this question about whether DNA or protein was the genetic material. The first experiment takes you back to 1928, Frederick Griffith. And Dr. Morgan talked about this a little bit yesterday in the intro to lab. I'd like to fill in a few gaps maybe along the way and kind of re-go over that experiment because it was a very important experiment. Another experiment we're going to talk about, which was kind of a variation of the Griffith experiment, took place in 1944, Avery McLeod McCarty. And then a very important experiment by Hershey and Chase in 1952. Together, these experiments really nailed down the answer to that question. What is the genetic material? Is it DNA or protein? Don't get too hung up on these particular dates. But I think it is important to understand the chronology, that this was the earliest experiment, this followed, and this followed. And then probably contextually, another thing that's really important is this all happened prior to 1953. Okay? This happened prior to the publication of Watson and Crick's paper. Because once the question was answered that DNA was the genetic information, then Watson and Crick rightfully said, well, if DNA is the genetic information and we want to understand it more, we've got to understand its structure. So in 1953, they published this paper outlining the structure of the DNA molecule. Okay, so let's go to Griffith for a second. Griffith was studying a disease, and the disease that he was studying was bacterial pneumonia. What happens in pneumonia? Your lungs fill up with fluid because of inflammation. Okay? Are all pneumonias caused by bacteria? No, there's several ways you can inflame your lungs. A bacterial infection is one, a viral infection is another, and then you can also get something called aspiration pneumonia, where you inhale food particles, for example, and they get into your lungs as opposed to going down the esophagus into your stomach. The causative agent that Griffiths was looking at in his study of bacterial pneumonia was an organism called Streptococcus pneumoniae. Okay? The species name pneumoniae obviously suggests that this organism can cause this particular disease. This bacteria is a pathogen, and it has the ability to cause disease. The ability to cause disease is called virulence. So this was a virulent organism, a disease-causing organism. You can actually tell something about the bacteria by its genus name, Streptococcus. Anybody know what you can tell about it? About the nature of these organisms? The shape of it, okay? What is a cocci? It's a circle. What's the other major morphological form? Bacillus, okay? You did some bacillus bacteria last semester when you did your mouthwash experiment. Strepto means chain, basically. So these organisms tend to form long chains. And it's relatively easy to identify these if you do a special type of staining process called a Gram stain. And Streptococcus stain as gram-positive organisms, and gram-positive organisms in this staining process stain purple. Did you guys do gram stains last semester? Okay. Sometimes you do, sometimes you don't. The other major type of bacteria, which are gram-negative bacteria, have a different staining. They stain pink. E. coli, which you're using in lab right now, is a gram-negative organism. Okay? So this gram stain is really easy to do, and if you were working with patients or mice that had pneumonia, you could collect a sample from their lungs and do this gram stain, and then you could, if you saw these little cocci that were purple in chains, pretty good bet it was the Streptococcus bacteria. Well, these particular bacteria exist in different forms that we call strains. And Griffith was working with 2 strains of this Streptococcus bacteria. One of them was designated the smooth strain, and the other strain was designated the rough strain. The rough bacteria, were avirulent, and the smooth bacteria were virulent. Remember what that word means, can cause disease. So these organisms could cause pneumonia, but these did not. Okay. Actually, these rough forms were a mutant. They had something that was lacking in them. These smooth forms were the wild type. That was the version that was normal, if you like, the non-mutant. Why did he call them rough and smooth? Well, if you look at these organisms growing on an agar plate, and you look at the colonies, they actually have different appearances. The smooth ones were kind of glistening and rounded, and the rough ones were a little bit more granular and a little bit more irregular. And if you take a little bit of sample from each of those colonies and make a slide out of it and look at the cells, they actually have a different appearance. These cocci in the smooth are surrounded by a structure called a capsule. Almost looks like a little halo. And these rough forms right here don't have the capsule. They're just the cell. Any idea why the encapsulated forms or the smooth forms were the ones that were disease causing?
Well, maybe the body has trouble killing the ones with the capsule.
It's absolutely the case, okay? These particular bacteria that have this capsule have kind of a protective shield around them. And it prevents one of the body's major defense systems, white blood cells, which can do the process of phagocytosis, from gobbling these things up. The rough ones, which lack the capsule, are readily attacked by those phagocytic cells and eliminated. So somehow the mutation that these have prevent them from making a capsule. These have the necessary genetic information to make this particular structure. So, here's the experiment Dr. Morton talked about yesterday a little bit. And Griffith did several experiments, some of which were a little bit bizarre. One of the experiments that he did was he took living smooth cells, which was a control experiment. You notice here that they have this capsule surrounding them, so they're able to survive in the mouse when they're injected. And when they're injected, the mouse develops pneumonia and dies. Lungs become all inflamed. Okay? When he took the rough strain, which lacked the capsule, and injected those into the mouse's immune system recognized these as being foreign cells, and its white blood cells gobbled those bacteria up before they could cause lung inflammation. So they survived. He then took some of these deadly smooth cells and he heated them. And the heating those cells destroyed the cells and killed them. And if the cells are killed, can they replicate when they're introduced into the mouse? And therefore, the mouse survives. But then for some reason, and I'm not sure why, curiosity maybe, he did this interesting experiment where he took a mixture of these and these, put them together. Now, remember, individually, both of these cells cannot cause death. But when he mixed those together and injected them into the mouse died. And furthermore, when he did an autopsy on the mouse and looked at the lung tissue, they were just filled with these smooth Streptococcus bacteria. Wait a minute, what's going on here? I mean, they were dead. Did somehow mixing these cells and these cells make these cells become alive again? Well, that didn't make much sense to Griffith. What he hypothesized was that there was something from these cells, some genetic information, that was released when they were killed with heat. And that information was taken up by these cells, and it changed them. It changed their genetic complement of information, and now these cells had the ability to make these capsules and to survive. So we changed the phenotype of these cells from the rough strain to the smooth strain. What's another word for changing the phenotype of a cell or organism? Which is what you did yesterday, if you had lab. This is called transformation. Okay? So Griffith's experiment was the first documented case of bacterial transformation where genetic information from one cell was taken up by another cell, and that cell's phenotype was then altered. Well, did this answer the question of whether the genetic information was DNA or protein? No. Okay. But it laid the groundwork for the next set of experiments. Who did those experiments? Avery, McLeod, and McCarty, about 16 years later, in 1944. They repeated the Griffith experiment with one major change. They heat-killed smooth cells, but then they went a step further. They took those cells and they isolated them into their 4 basic cellular types of molecules. What are the 4 basic molecules that make up all cells? Carbohydrates, lipids, proteins, nucleic acids. Okay? So they took these heat-killed cells and separated out those classes of molecules. And then they mixed living cells, which were rough, with one of those 4 extracts from the dead cells. Okay? So living cells were mixed with the DNA fraction. Living cells were mixed with the protein, with the carbohydrate, and with the lipid fraction. And only one of those 4 mixtures caused transformation. Which fraction was it? It was the DNA fraction, or the nucleic acid fraction. Okay? That mixture of molecules included both DNA and RNA. So, that 1944 experiment strongly suggested that it was the nucleic acid portion of the chromosome which actually encoded the genetic information. The instructions for how to make those capsules. Okay? These cells didn't have that information before the transformation. But when you added DNA from cells that had that information, these were now transformed and could do it. So that was pretty compelling evidence. But those people who really believed in protein as genetic information were a pretty tenacious lot. And they didn't buy into this easily. The real nail in the coffin came 8 years later in 1952 when Hershey and Chase did a very different experiment to try to answer this same question. What's your genetic information? Is it DNA or protein? Hershey and Chase didn't work with bacteria. They instead worked with a virus. And they worked with a particular type of virus called a T phage. Anytime you see the word phage or phage, you immediately should understand something. This is a virus that infects a bacterial cell. Okay? Not a human cell, but a bacterial cell. T phages were very interesting virus. They had a pretty complicated morphology. There was a region of the virus called the head. There was a little collar that connected the head to this base plate. And then there were structures protruding from this base plate called the tail fibers. This whole thing right here is called a capsid. The whole thing is a capsid. Now, not all viruses look like this. Some viruses have a capsid that's completely round. Some viruses have a capsid that is tubular. But these T phages had this very kind of bizarre, interesting looking capsid structure. And here's what a T phage looks like in an electron micrograph. Okay, so you can see the head, the collar, base plate, tail fibers. This capsid is made entirely of protein. The little tail fibers right here dock on to the surface of a cell and allow that virus to attach to the cell. And if a virus can't attach, it can't infect the cell. Okay? So these tail fibers are for attachment. And they attach to certain receptors on the surface of the cell. If I exposed you to these T phage virus, would they make you sick? Nope, they are specific for bacterial cells. Why won't they infect your cells? Because these tail fibers do not match receptors you have on the surface of your cell. And if they can't attach, they can't infect. Now, coiled up in this little head region is the second part of a virus. It's nucleic acid. And the nucleic acid of the T phage is DNA. We're going to talk about virus later in the semester, and we're going to see there are other kinds of virus that have other kinds of nucleic acid. Some have RNA as their nucleic acid, but these have DNA. When a virus infects a cell, what does it do to it?
It inserts its DNA into the bacteria's DNA.
Okay, it inserts its genetic material. I'm not going to call it DNA yet, okay? But that's true. Inside the cell, and it takes over the cell. It hijacks the cell. And it reprograms the cell. And what's the cell dedicated to doing now once it's reprogrammed? Make new virus. So it turns that cell into a virus factory. And then at some point in time, the cell fills up with new virus and it bursts, and all these new viruses are released, and they can then infect new cells. So viral infections, especially in multicellular organisms, can really get out of control. And oftentimes, once you start damaging a significant amount of cells, you have some consequences, like a developing cold, for example, which I think I'm getting. So viruses are very interesting. Are they cells? No. Because they only have two of the four components that cells are made up of. They have DNA, some type of nucleic acid, and they have protein. They don't have carbohydrate, they don't have lipid. Can they replicate? Yes, but only if they're inside of another living cell. So Hershey and Chase reasoned this way. Whatever these cells introduce into the cytoplasm of the bacteria they infect is what is going to reprogram the cell. So that must be their genetic information. So they said, okay, what's getting into the cell during this infectious process? Is it the nucleic acid portion, or is it the protein portion? So they did a relatively simple but pretty elegant experiment where they labeled atoms selectively in DNA and protein. And they used two isotopes to do this experiment. both of which are radioactive, so their presence can be detected. Okay? You have a Geiger counter or some kind of other instrument that can measure radiation. They used P32 and S35. Which one did they label the DNA with? Does protein have phosphorus in it? A DNA does. It's got phosphate groups. So they produced radioactively labeled DNA P32. Do proteins have sulfur in them? Some do. Okay? There are several amino acids that have sulfur as one of their R functional groups. They're called sulfhydryl groups. So the S35 labeled the protein. So basically what they did was they made these labeled T phages where the DNA was labeled with one thing and the protein was labeled with another, and then infected bacterial cells and then saw what got into the cytoplasm of that cell. Did the phosphorus get in or the sulfur get in? And when they did that experiment, which is outlined in this particular slide right here, they found out that the proteins stayed outside of the cell when infection occurred, but the DNA got inside the cell when the infection occurred. And genetic information has to get inside the cell in order for that virus to then be attacked and killed. It was very easy for them to determine whether the cells were dead or living, the bacterial cells were dead or living, and by following this distribution of radioisotope, they were able to determine what got into the cell and therefore what was the genetic information. This is sometimes called the Waring blender or blender experiment because one of the steps in this particular experiment involved taking these cells that had been infected and stirring them up really fast, and that knocked off these little capsids that had been adhering to the outside of the cell. And then they could measure radioactivity in both the supernatant, the liquid, and in the pellet, which represented the cells. So this very clearly demonstrated and reinforced the 1944 experiment by Avery McLeod and McCarty that DNA was indeed the genetic information. 1952. Watson and Crick read this paper and they went, okay, DNA is the molecule to understand if you want to understand molecular genetics. So they began their research. Watson and Crick were interesting choices to really be the makers of history in molecular biology. James Watson was an American-born and trained PhD. He really wasn't a molecular biologist, and in one of his books he really explains that he didn't know a whole lot about biochemistry. He didn't even really know that much about the structure of nucleotides. We did know at the time that DNA was made of these four nucleotides, A, T, G, and C, but no one knew how they were put together to form the functioning DNA molecule. Watson did a postdoc in England at Cambridge, and there he met a physicist, Francis Crick, who was also studying at Cambridge, and they both decided we want to make our mark in biology, and therefore we're going to study this DNA stuff and try to figure out what the structure of DNA is. Because they understood structure determines function. If we understand what the structure is, then maybe we can answer some of those questions of function, like how does this molecule replicate? Or how does this molecule encode genetic information? This is a picture of Watson and Crick. James Watson on the left, Francis Crick on the right. And one of the things that they did that was kind of unique for the time was they were involved in model building. And they were ultimately trying to come up with a model, which we have a plastic representation of here now, of what the DNA structure actually was like. To construct their model, they had to use these ring stands and clamps and cut out pieces of wire and tin, which represented the bonds and the four bases, A, T, G, and C, and try to put them together so that the model explained physical evidence about DNA. So the model had to be predictive. It had to accommodate what was known at the time about DNA and what they were learning as this research was going on. What did they have to know? Well, one thing that this model had to account for was something called Chargaff's rules. And this guy Chargaff had done some analysis of DNA. And he had found out that no matter what DNA you look at, from what source, that the amounts of these adenine nucleotides always equal, or very closely equals, the amount of thymine nucleotides, and the amount of guanine nucleotides also equal the amount of cytosine nucleotides. So A equals T, G equals C. The other set of information about the physical characteristics of DNA came from a technique that was being done at Cambridge at the same time called X-ray diffraction. And X-ray diffraction was carried out at Cambridge by Maurice Wilkins and Rosalind Franklin. And Rosalind Franklin is a very interesting story, and we'll allude to a little bit of her story as we talk about this. If you're interested in some optional points, Natalie has a video downstairs that you can watch and answer some questions about, called The Secret of Photo 51, which describes the contributions that Franklin made to the discovery of the structure of DNA and how she was shafted and basically didn't get any recognition. Watson and Crick won the Nobel Prize, and Franklin was totally left out. Why? Because she was a woman. In 1953, women scientists were definitely second class. And then we'll also talk a little bit about Linus Pauling and the role that Linus Pauling played in this kind of race to discover the structure of DNA. And this indeed was a race, okay? This was the prize of biology at the time. And the groups working on this fully understood that fame and fortune would come to the people who published the results of this group. So we had this British group, Watson and Crick, supplemented by Wilkins and Franklin working on it. And then we had the American biochemist Pauling, who already had a Nobel Prize under his belt for discovering three-dimensional structure of proteins, working on it in California. So we'll continue the story on Thursday. So start reading the chapter on DNA if you haven't.